Welcome back, kids and coaches. And today we are getting into learning outcome 4, which is looking at the mitochondria. And we're going to be talking about oxidative phosphorylation here and aerobic glycolysis. So, what this is going to cover is really the role of mitochondria and how we use oxygen to actually make ATP. So, this slide may be a little bit familiar here to you, but the mitochondria is colloquially known as the powerhouse of the cell. And rightfully so, this is where the electron transport chain is located. So this is where the majority of your ATP is actually generated, is in the mitochondria of your cells. Now, some cells will have very few mitochondria in them. Some will have a lot of them in them. For example, some cells may have a single mitochondrion. Others, such as say like the liver, have up to roughly 2,000 mitochondria per liver cell. So mitochondria are present when in an abundance when they're necessary for the function of the surrounding tissue. So the more energy tissue needs, the more mitochondria that are going to be present in each cell of that tissue. Now, one of the things that we'll see later on in the course is that when we are seeing adaptations to exercise, one of the things that normally happens is as you become more well trained, both aerobically and anaerobically, you're going to get an influx of mitochondria systemically. Typically, this is going to result in an increase in mitochondria in your skeletal muscle and your liver. Now, if you look at this picture here, this gives you an idea of what the mitochondria actually looks like under an electron microscope. So when we see the different animations or drawings of it, this doesn't look all that different. These here, see all those different lines there. This is part of the inner membrane, and this is really just the structure in which we see the uh, the electron transport chain occurring. Now, just outside of this, so just outside of the mitochondria, this is the cytosol. That's where glycolysis is going to occur. So that just occurs just on the outside of the mitochondrial wall. One other thing to note with mitochondria is it has its own DNA. So you have mitochondrial DNA. This is separate from, say, like the DNA you have making up your genetics or your, say, your chromosomes. Your mitochondrial DNA is purely maternally inherited, meaning you only get mitochondrial DNA from your biological mother. You do not get any mitochondrial DNA from your biological father. This means that you have to be related to every single one of your siblings, pending that you all have the same biological mother. I had a high school biology teacher once say that you could theoretically be biologically unrelated to a sibling if each of you had two separate sets of chromosomes, meaning the 23 chromosomes you got, say, from your mother and the 23 from your father were the completely separate set you would get if your sibling had the complete opposite set of their 46, which, if you really think about it, is kind of nonsense because there's no way you can't be biologically related to a full sibling. But if you have the same mother, you have to share the same mitochondrial DNA. The only way that you can get mitochondrial DNA is through maternal inheritance. Now, when we look at the differences here between aerobic and anaerobic oxidation or anaerobic and aerobic metabolism, we're simply talking about the difference here in how we use oxygen or the lack thereof. When we have anaerobic glycolysis. You're taking glucose, and then at the last step here, you'll see that you'll get ATP, you get NADH, and you get pyruvate. That's the same on both sides of this. Where the differences occur is what happens after we get the pyruvate. When pyruvate is generated and we have oxygen, it gets converted into acetyl-CoA. Acetyl-CoA, as we know, is going to get transferred to the citric acid cycle or the Krebs cycle, and then that ends up going to the electron transport chain, and that gives us ATP. If we don't have oxygen, so if oxygen is not present, our pyruvate enters what is known as the lactic acid cycle or lactic acid fermentation. Here, pyruvate gets converted into lactate or lactic acid. If you notice, there's no sending this to acetyl-CoA. It doesn't happen. What happens here is that we have to take our lactate and then we have to then further convert this through a lactate dehydrogenase enzyme before this can then be converted back into pyruvate, into acetyl-CoA, and then into the citric acid cycle. So if you don't have oxygen, this takes a lot longer to recycle than if you do have oxygen. So the key here is if we have oxygen, we can limit lactic acid buildup because we can turn pyruvate into acetyl-CoA. Acetyl-CoA is great because this is a precursor to ATP. Lactate is not. If you have high levels of lactate in your body, you are going to fatigue. This is what accounts for, say, the burn you feel when you're exercising, especially when you're doing resistance training. The more lactic acid you get built up, the quicker you fatigue, and the more you feel that muscle burn. If you are able to oxidize pyruvate, you're not going to get that lactic acid buildup. You will have less fatigue, and you'll be able to exercise for a longer duration. Typically, the higher the intensity of the exercise, the more lactic acid production. So as intensity increases, lactic acid also increases. As intensity decreases, lactic acid decreases. Because when we have low intensity, we are able to oxidize the pyruvate into acetyl-CoA. This is also why recovery days are so important for your regular exercise routine. If you are exercising at full go, like 100-ish% of your heart rate reserve or close to your VO2 max, your body is not going to be able to effectively get rid of lactate on a regular basis. If you have a recovery day where you have low intensity workouts, think of like, say, a low intensity cardio day, that's going to actually help with converting this pyruvate into acetyl-CoA, or it's going to help with getting rid of the excess lactic acid buildup and being able to eventually convert that back into acetyl-CoA. Now, if you have too much lactic acid in your body, you have a condition known as lactic acidosis. What happens here is that this is an indication that your body has a problem with your oxidative metabolism. Now, we'll get into that here in a second. When you are actually starting this process, so first off, you're going to take glycogen. You're going to convert that into glucose. So you're going to break that down into glucose. Then it's going to enter glycolysis. You have two types of glycogen stores. Glycogen either comes from muscle or it comes from your liver. These two stores have very different uses. If you get glycogen from muscle, you are going to use this to get ATP. If you get glycogen from your liver, this is used for blood glucose regulation. One of the hormones we'll talk about when we get into our unit on the endocrine system is called glucagon. When glucagon is present, your body says that you have low levels of blood glucose and we need to release more glycogen to raise our blood glucose levels. So glucagon signals to the liver to release glycogen, which then gets converted into blood glucose to regulate our blood glucose levels. When you are getting ATP, you are getting glycogen from your muscles. The other benefit to that is that when you get glycogen from your muscles, the ATP is already where it needs to be. It doesn't have to be transported from the liver to the muscle you're working. It's already there. So your body has this nice little checks and balances system where it can immediately send ATP right where it needs to be because it's already located there. Now, we've talked about the Cori cycle before, but this is the process of how we take lactate and turn it back into glucose. The Cori cycle prevents lactic acidosis from occurring. And again, lactic acidosis just means that you have high blood lactate levels, but it's high blood lactate levels that aren't resolving themselves. This means that you have a problem with your aerobic metabolism or your oxidative metabolism. Something's going wrong there. You're not able to use oxygen to make ATP. And as we know, glucose will be converted into ATP via anaerobic glycolysis, but it only gives you 2 ATP or a net of 2 ATP per glucose molecule. When you break down a molecule of glucose anaerobically, you get only about 5% of the ATP that glucose molecule has. Glucose should have roughly 38 ATP in it. If you cannot use oxygen, you're only getting about 5% out of that, which is not a lot. It's very, very inefficient. However, anaerobic glycolysis will occur about 100 times faster than aerobic glycolysis. So imagine you're riding a bicycle and you have a bicycle that has 10 gears on it. If you're in first gear, it's the lightest gear. You can pedal the fastest, but you go the slowest. 10th gear, you pedal the slowest because there's the highest amount of resistance there, but you go the fastest. That's how fundamentally we have the difference between aerobic and anaerobic glycolysis. Anaerobic glycolysis is gear 1. Aerobic glycolysis is gear 10. You cannot start at gear 10. If you were at a dead stop and tried to go to the hardest gear you can go to, you wouldn't go. Imagine you're in your car and you have a manual transmission. You always start your car out in gear 1. If you start your car out in gear 6, you're not going to go anywhere. Your car's going to stall out. You got to build up to it. Well, you start with gear 1. It's the fastest that you're going to go, but it's the fastest you're going to pedal or the fastest your engine's going to go, but it's going to be the slowest for you in terms of how fast you go. This is because you have the smallest gear shaft with there, whereas with gear 10, you have the biggest gear shaft. You need to start with gear 1 before you make it to gear 10. You have to start with anaerobic before you go to aerobic because it takes longer for oxygen to work. Now, when you go into the Cori cycle, what's happening here is that we start with glucose, we go through glycolysis, and this happens here in the muscle. After we go through this glycolysis, and again, we're saying this is anaerobic glycolysis here, we end up with lactate. We can't use lactate. Lactate has to be recycled before it can be reused. So what happens is the Cori cycle takes this lactate, it sends it over to the liver, and you reconvert that to pyruvate via lactase dehydrogenase. So you may see that as LDH or LD. But the problem with this is gluconeogenesis, is that it costs 6 ATP. What's happening here is that when you don't have oxygen, you get a net 2 ATP initially, but then you have to repay that with interest to convert your waste products back into glucose. So anaerobic glycolysis, one cycle of it, actually costs you 4 ATP. Now, what does that actually mean? Well, in terms of energy balance, it means you use more energy than you produce. That means that if you were exercising, you're exercising at a high intensity. When you exercise at a high intensity, you end up burning more calories. This is partially the process in which that occurs. So as we generate more glucose through non-glucose factors, we have to pay ATP to do this because this is anabolic. When something's anabolic, it builds up. When I do this here with glycolysis, it's catabolic, or it breaks down. So what's happening with the Cori cycle is that we are acknowledging it's going to cost us more ATP to fix our waste problem than what we actually produce. But in turn, we don't get lactic acidosis, or we don't get blood lactate to accumulate. This is why if you have even a hard exercise or a hard exercise session, and then you go and you say do a recovery the next day, or even if you do nothing the next day, most all of your blood lactate will reconvert into pyruvate and then into glucose, potentially stored as glycogen then, because of the Cori cycle. You spend more ATP doing this, but it results in a normalization of your blood lactate levels. If it weren't for this, you would go through lactic acidosis. So here's a little bonus for you. One of the things that can result of having a mitochondrial defect is that you have issues with your oxidative system, or your oxidative phosphorylation. There we go. One of the big conditions that can happen as a result of this is called the Mila Syndrome, which is what I have. Mila stands for mitochondrial encephalopathy with lactic acidosis and stroke-like episodes. So let's break this down from a fundamental standpoint. Mitochondrial has to do with the mitochondria. Encephalopathy If you have encephalopathy, there we go, that means that you're going to have issues here with your cell pathways. We talked about lactic acidosis. So lactic acidosis is the buildup of um, lactic acid without being able to reuse it. And then stroke-like episodes. So like you would see strokes, someone have a stroke. This is stroke-like episodes. So the reason it's a stroke-like episode is because they can potentially resolve themselves. All mitochondrial diseases occur from your maternal inheritance. This means that if you have a mother that doesn't have any mitochondrial disease, all of her offspring cannot have mitochondrial diseases unless there's some other extenuating factor that would cause that. So for example, you're exposed to like some sort of ionizing radiation or something like that, that could potentially cause that to happen. But you can only get a mitochondrial disease from your mother. With Milos, the big problem is right here. Now, this is a gram stain of your mitochondria. Your mitochondria should look like this. When you see them looking like this, or where these red blobs are, that's what are known as ragged red fibers. What happens here is the mitochondria themselves are considered diseased and they don't work properly. If you had all of your mitochondria like this, you would die because you cannot make ATP then through oxidative phosphorylation. When you are symptomatic for a mitochondrial disease, you typically have about a 60% mutation rate, meaning that 60% of all of your mitochondria look like this. They are all defective. When this happens, you build up lactic acid and you cannot reuse it, because the only way you truly get rid of lactic acid is through oxidation. If you can't oxidize it, it doesn't get broken down, and continuously pyruvate will convert to lactate and build up lactic acid in your body. Ultimately, if you have too high of lactic acid levels in your body, it'll actually break down your muscles. So you would actually see your muscles degenerate really in front of you. Unfortunately, there's really no known cure for this, and there's not really a whole lot of treatment that happens. So, knock on wood that I'm kind of living on borrowed time here. But with that being said, I hope that wasn't too big of a downer for you. And hopefully now you have a little bit better of appreciation and understanding of your mitochondria and how we have a difference here between aerobic and anaerobic glycolysis. And until next time, I will see you all later.